infectiousness. And we've also developed a whole lot of other ways to prevent HIV in addition to safe sex. I mean, most people know that a condom will just prevent transmission of HIV, but we know that human beings are human beings and they just won't use condoms all the time. And there's been a real revolution in the field of what we call biomedical prevention, ways that we can prevent HIV other than behavioural modification. And they include taking anti-HIV drugs. So if an HIV negative person takes an anti-HIV drug each day, what we call pre-exposure prophylaxis or PrEP, it reduces the chance of being infected by 40%. It's a bit like taking the pill to prevent pregnancy, but this is quite expensive. It's at $26 a day, so implementing it will be quite difficult. We know that circumcision dramatically reduces your risk of acquisition of HIV, and in fact reduces the risk of a male from acquiring HIV by 70%. And that's largely because of the infection-fighting cells that sit on the inner aspect of the foreskins, cell called dendritic cells, where it's very enriched on the inner aspect of the foreskin. If you remove it, you remove the risk of acquiring HIV. And then probably one of the most important discoveries, because this is one of the few female-driven approaches to prevention, is to use what's called a microbicide or a gel that contains an anti-HIV drug, in this case a drug called tenofovir, which applied before and after sex reduces the risk of acquisition of HIV by 40%. So there have been some amazing, amazing victories in how we use drugs, how we can treat people who have the disease, strategies we can use for uninfected people to prevent them getting the disease. But they're all quite expensive. All the drugs, even with commitment from drug companies, governments, generation of generic drugs, this is still an expensive strategy. So there are a number of challenges that still remain. And the first one is that even though people get much better when you treat them for HIV, they stop dying of AIDS, and they certainly live longer, there is still significant morbidity that persists in patients on treatment. This is largely due to a combination of some toxicity of the drugs themselves, residual immune activation. So if anything, when a person is infected with HIV, even though it erodes their immune system, part of the problem is that the immune system is too vigorous in its response to the virus, and it remains activated even in people on treatment. And the HIV is still there even when you're taking drugs and can't measure it in blood. And as a result of this, what we are seeing more and more in patients with HIV on treatment is a range of diseases that are familiar to us because we largely see them in the setting of ageing, so cardiovascular disease, metabolic disorders, brain disorders. And this is largely thought to be due to ongoing inflammation that persists in patients on treatment. We don't have an effective vaccine. We know a lot about how the immune system responds to HIV, what we think we would like to see from an effective vaccine. We think we'd like to be able to induce a good antibody response, induce a good T-cell response. But this, out of many, many vaccines that have been tested, this is the only vaccine that has shown any benefit. And this vaccine was a vaccine designed to induce an antibody response and induce a T-cell response. It was given to 16,000, the study was done in 16,000 people, again, placebo-controlled, meaning you flip a coin, you give half placebo and half the vaccine. And the chance of getting HIV infection was reduced by about 30%. So it looks like it worked. The problem is we don't know why it worked. The investigators thought that it was going to work because this vaccine would induce antibodies and induce T-cells, but that doesn't seem to be the case. And there's a lot of research now trying to understand why and how this vaccine worked. And some of that research being done right here at University of Melbourne by Stephen Kent. So a lot of unanswered questions, and we still don't have a vaccine. And it's very difficult to be talking about the great successes of antiretroviral therapy without talking about the money that we need to fund it. So we have 6.5 million people currently on treatment in the developing world, and it costs about $15 billion a year to fund. The goal is to get about 15 million people on treatment, which would be about 80% of people who need it. And the estimated cost is around $23 billion. It's hard to understand what $23 billion means, but it's about half the foreign aid budget of the US. So it's even though we know that keeping people on treatment lifelong could have a major effect on the epidemic, funding this is going to be very difficult. So, what are the big challenges in the road ahead? Well, without a doubt, the major scientific challenges are still to find a vaccine and to find a cure. Because if we found a cure for HIV, we wouldn't need to be treating people for 60 years. Perhaps we could treat people for 3 or 5 years and then safely stop the drugs if we managed to, if the person could then control the virus themselves, or if we could eliminate HIV altogether. And I'm going to speak a little bit more in detail about some of the work that we've been doing and what others have been doing in trying to find a cure for HIV. And to understand why we don't have a cure and why antiretroviral therapy doesn't eliminate every last scrap of HIV, I just need to explain to you briefly about how HIV replicates in T cells. So we heard before that when a CD4 T cell or a T cell makes a response to a foreign antigen, it actually gets activated, and it's part of its role in clearing away a foreign pathogen. And that's exactly the cell that HIV preferentially infects and replicates in. It gets into that cell, it produces lots of virus particles and kills the target cell. If you're on treatment, then the viruses are unable to go on to infect the next activated T cell, and that's how combination antiretroviral therapy works. But in a resting T cell, the virus has this amazing evasion capacity. It is able to enter the cell, integrate into the host DNA, and basically hide there. It can come out at any time if this cell becomes activated or something else activates the virus. And this is what we mean by latency. And many viruses have the capacity to establish latency. But this is really the main reason. These latently infected cells don't budge an inch when you put someone on treatment. The virus is sitting there hiding inside the DNA and will stay inside the DNA for the lifespan of that cell. And we've been very interested in studying how a virus gets into resting T cells, how it stays there, and how you might be able to one day push it out of those resting T cells. And we've done a lot of work in understanding infection of resting T cells, because if you take a virus and try to infect a resting T cell, you actually can't do it in the laboratory. But we found several years ago that there's a number of important proteins or chemokines, special proteins that guide T cells around the body that facilitate infection of a resting T cell. Now, latently infected cells in patients on treatment are very rare. On average, it's about 1 in a million cells are latently infected. And unfortunately, they're not painted light pink, or they don't express any foreign antigen. So it's very hard to actually identify a latently infected cell, and therefore very hard to study latency when you take blood from an infected person. So we use our in vitro model of we use our in vitro model of latency to understand factors that control or will reverse latency. So one of the ideas that people are pursuing at the moment to eliminate latency is sort of counterintuitive. It's the idea is that if we could activate that virus or get it to start making copies of itself, making it visible perhaps to the immune system, or even producing virus, this could be safely done in a person that's already on antiretroviral therapy, because any virus that's coming out of the cell in a person that's on treatment won't go on to infect new cells. And that latently infected cell would either die by the virus coming out or will become visible to the immune system. So the idea is that you're actually waking up the virus from latency, pushing it out of its hiding spot, and eventually killing that target cell. And we've been using this model that we have that we can study latency in the laboratory to screen drugs that activate latent infection. And we know that latent infection has been activated because then we can measure the virus in the experiment. And there's a number of drugs that do this. So one group of drugs are called histone deacetylase inhibitors. These are drugs that turn genes on and off and are used for the treatment of cancer. But they actually turn on HIV genes very effectively, at least in the laboratory. And this one drug, Saha, or verinostat, is a drug that's already licensed for the treatment of cancer, and therefore was the logical drug to choose to then test in people on treatment to see whether it does the same thing in people. We're currently doing a clinical trial of this drug at the Alfred in patients who are HIV positive on antiretroviral treatment. They receive 14 days of the drug, and we're trying to work out whether we can wake the virus up from latency. The other approach to curing HIV is quite different, and it comes from the success story of this man here. His name's Timothy Brown. And Timothy Brown is the only man that's ever been cured of HIV. And he was cured because he had leukemia and he needed a bone marrow transplant. And his clever physician in Germany decided to give him a bone marrow transplant from a person that he knew was resistant to HIV. About 1 to 2% of people are resistant to HIV because they don't have a specific receptor called CCR5 that the virus needs to get into the cell. So this man received a bone marrow transplant from a donor who lacked CCR5. And shortly after his transplant, his doctor stopped treatment and no virus rebounded. No virus could be found in blood. No virus could be found anywhere in this man now for about 4 years. So transplantation, as you heard from Glenn, is a very, very invasive, expensive, dangerous procedure. He had to have it because he had leukemia. But now there's a whole area of research trying to work out maybe we can mimic what happened to that man in a slightly gentler form. And the approach is being tested on this man on the right, whose name is Matt Sharp, who's participating in some gene therapy studies where we use gene scissors to make cells resistant to HIV. So you can very specifically target these gene scissors to remove the CCR5 gene from T cells. You can do that in the laboratory and then return those CCR5 negative T cells to patients. It's been done now in about 20 patients. Those CCR5 negative cells survive. And one day we may learn, there's a lot of work going on still whether they could one day eliminate HIV. So, will we see the end of AIDS? Well, to see the end of AIDS, I think we're going to need 3 things. We will need to seek, test and treat everyone who's HIV infected. And if we could actually do this or fund this strategy, technically this could lead to the end of HIV because we could totally reduce transmission. But the feasibility of this, particularly in the current financial environment, is, I think, highly unlikely given the cost and commitment this is going to take. I think we still need to invest significantly in the science to find a vaccine. We have got many other prevention strategies, but they're all a bit complicated and they're all expensive and they require ongoing commitment from that person. A vaccine is really the only simple, cheap way we'll ever be able to universally protect people from HIV. And finally, even if we found a vaccine tomorrow and eliminated every new infection, we still have 33 million people infected with HIV, and we need to find a way that they can safely stop their medications. So, will we see the end of AIDS? I think if in the next 30 years we see as much progress as we've seen in the last 30 years, it just might be possible. And I'd just like to thank a number of people that work with me at Monash on HIV latency, particularly Gabby Corey, who's been involved in the Immunology day-to-day, colleagues at the Alfred Hospital doing the clinical trial of verinostat, and a number of funders that support my work. Thank you.